In our final lesson regarding buffers, let's take a look at some naturally occurring buffers and see how they compare. We also want to briefly review how buffers are prepared in the lab. Recall that the buffering capacity relates to the actual amount of acid and conjugate base present in the buffer solution. The higher the amounts of each, the higher the buffering capacity. To make a buffer in the lab, we might start with the acid form and use a strong base like sodium hydroxide to convert some of the acid to conjugate base up to our target pH. Instead, we could also begin with the conjugate base and add a strong acid like HCl to convert some to the acid form, thereby making a solution containing both forms. There are two main buffering systems in the human body. A phosphate buffer is the principal one within the cell, and the relevant pKa here is 7.20. In the blood, a buffering system is employed that is based on the dissociation of carbonic acid, which has a pKa of 6.37. As we will see much later in this series, carbon dioxide is the waste product associated with aerobic metabolism. It can dissolve in water to give carbonic acid, as illustrated in the first chemical equilibrium here. The pKa of carbonic acid is approximately 6.4, while the pH of blood is around 7.4. This means that carbonic acid will readily dissociate to its conjugate base form, bicarbonate. Although this process occurs spontaneously, the conversion of CO2 to bicarb is greatly enhanced by the enzyme carbonic anhydrase. These are two examples of buffer systems that operate in the living cell, that is to say, in vivo. But we might also need a buffer system to function in vitro, outside the body, in the lab. Non-biological buffers are often Zwitter ions, that is to say, they are compounds that are twin ions, that have both a positive and negative charge. These are buffers like the ones listed in the table here, which are less likely to interfere in biochemical reactions than other naturally occurring buffers. Finally, let's emphasize that all living systems operate within a narrow pH range, and therefore all must be buffered in some way. In other words, in biology, pH always matters. This means that we will need to apply the principles related to pH and pKa to almost all of the molecules and systems that we encounter in our study of biochemistry.